In this video, we're going to go ahead and move on in the 5.2 notes. We're going to look at topic 3. In topic 3, we're going to talk about something called gene linkage. And so in this video, I'll go over first of all, how this was discovered and then what this is, and ultimately how this thing called gene linkage is going to affect or change the way that traits are inherited sometimes, and how they're passed on from the parents to the offspring, and make it, this is going to make it a little, certain traits get passed on. This is going to cause certain traits to get passed on in different ways that vary sometimes a lot from what Mendel would have predicted. And we'll try to understand why that's happening. So the short answer, or the short description right now, is that gene linkage is when you have genes located on the same chromosome. So when you have two different genes that are coincidentally on the same chromosome, which happens pretty often actually, because a chromosome is going to have hundreds of genes on it. So by coincidence, it's not too surprising that every now and then you might be looking at two genes that are on the same chromosome. That's called gene linkage. And when they're on the same chromosome, that's going to mess things up a little bit in terms of how those alleles for those genes are going to be passed on. So that's what we're going to look at. So we're going to start by looking at how this was discovered, again, by this guy named Thomas Hunt Morgan. So I talked about him in the last video in topic 2, and how he discovered sex linkage, how genes are sometimes, he figured out that some genes are located on the X chromosome. He's also going to discover that this whole thing about gene linkage, that sometimes there's genes located on the same chromosome, and that's going to cause things to happen a little weird, or a little not unexpectedly sometimes in these crosses. And so in these Drosophila flies, he was doing lots of different crosses and tracking lots of different traits in these fruit flies. And so one famous popular type of cross he was doing was with dihybrid flies that were heterozygous for two traits, and crossing them with homozygous recessive flies for both those traits. And so it's a lot like Mendel, actually. Mendel was also, in a lot of his pea plants, he was crossing pea plants and looking at multiple traits at the same time, and he was looking at dihybrids and crossing them with other types of pea plants. And so this guy, Thomas Hunt Morgan, he's going to do something similar with these fruit flies. I'll walk you guys through an example of what the crosses look like that he was doing. So let's do an example with two genes, and we'll look at the gene in these Drosophila flies. Let's look at two genes, one gene for the body color of the fly, and then which there's two versions that exist for body color in these flies. There's black and there's gray. Most of the flies are gray, and gray is actually dominant. It's what we call the wild type. That's the normal allele, the normal version of body color, and it's dominant to black. And then the wings, most of the flies have these nice normal functioning wings, but there's some flies who have this mutant version of the wings that are called vestigial wings. They do not work very well. And so the normal wings are dominant to the vestigial wings. And so gray is dominant and normal wings is dominant. But there's these mutant versions. So those are the wild type versions, the normal versions. And then there's these mutant versions, which are having like a black body. That's a mutant version of body color. And these vestigial wings, that's a mutant version. And so for these traits, we're going to use the letters B and V to represent the mutant versions of these traits. But they're recessive. So lowercase b, just know that stands for black, having a black body. And lowercase v stands for having these vestigial wings. And if you have an uppercase B, that is going to actually stand for having a gray body. So just be careful. Uppercase B is going to represent the normal phenotype, which is gray. And the uppercase V is going to be for normal wings. So even though we're using B and V, we're using B and V because a lot of times for the mutant version, if we're looking at a trait that has a mutant version, we use the letter that represents the mutant version. But since it's recessive, those are lowercase. Anyway, so he's going to do a cross basically just like this, where we're going to take a fly that's completely recessive for both those traits. So this fly has a black body and vestigial wings, which means they're homozygous recessive for both, because those are recessive traits. And we're going to cross it with a dihybrid fly. This fly is heterozygous for body color. It has a gray allele and a black allele. Which means it's going to have a gray body because gray is dominant, and it's going to have a dominant allele for wings, for normal wings, and a recessive allele for vestigial wings. But phenotypically, it's going to have normal wings because normal wings is dominant. So he's basically taking a fly like this and crossing it with a fly like that. Now, according to Mendel, like how this would play out, if we were to go ahead and set up a cross for this and make a Punnett square, you would first figure out, okay, what combination of alleles could this fly pass on, and what combination of alleles could this fly pass on? They have to pass on an allele for body color and an allele for their wings. And so this fly, there's actually, since it's a dihybrid, there's 4 combinations of alleles this fly could pass on. It could pass on a dominant and a dominant allele for each of those genes, or a recessive and a recessive allele, or a recessive and a dominant allele, or a dominant recessive allele, right? So we've done this a lot. According to the law of independent assortment of chromosomes, or of alleles, you should be able to give any combination of these alleles. As long as you're giving one allele for each gene, any combination should be equally possible, right? So that's all the different types of, let's say this is dad. That's all the different types of sperm dad is making. 1/4 of his sperm will have those 2 letters, those 2 letters, those 2 letters, those 2 letters. And then mom, the other fly, there's only one combination she gives. Since she's homozygous recessive, she's going to have to always give a recessive B and a recessive V, the allele for black body and vestigial wings, because that's all she's got. So for this cross, you actually only need a 1/4 Punnett square. It's pretty simple. This Punnett square here, and I already filled it in for you. So there's really 4 outcomes here that are possible, depending on if it's one of these gametes here meeting with this type of gamete that the other parent is making. You end up with these flies. Now, among these possible offspring, some of them have what we call the parental phenotype. Parental phenotype is refers to when there's an offspring produced who has the same combination of phenotypes as one of the parents. So like this fly is going to end up gray with normal wings. Well, that's just like dad. Dad also was gray with normal wings. So that's the same combination that dad had. We call that the parental phenotype. Or like this fly is black with vestigial wings. That's just like mom. Mom also has a black body with vestigial wings. So that's a parental phenotype. It's the same combination as one of the parents. So these two we call parental phenotypes, which happens, like sometimes you end up coincidentally with the same 2 combinations of phenotypes that your mom or your dad have. But then you can see that it's also possible, according to this Punnett square, it should be possible that you end up with these new combinations that mom and dad did not have. Like this fly has a black body, but with normal wings, if it's this sperm and egg cell that combine. And that's not the same combination that dad or mom have. It's a recombinant combination. It's a new combination. So we call that a recombinant phenotype. And this one too has a gray body with vestigial wings. We call that a recombinant phenotype. It's not the same combination as mom's dad. Anyway, according to this Punnett square, any of these should be possible. And not only that, they should all be equally possible. There should be a 1/4 chance that any of these flies are produced, because all of this person's gametes should be this, and this person's gametes, if the law of independent assortment is true, and you can give any B with any V, and any combination is possible, it should be this, 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 or this, or that. And so here's the expected outcome. This is what we would predict to happen if everything's playing out according to how Mendel would have predicted. Now, the actual outcome, which I'll show you on the next slide, was not this. It wasn't 25%, 25%, 25%, 25%. And so when Morgan did this cross, he produced hundreds and hundreds of offspring from crosses like this between these 2 types of parents. And here was his actual results. It was not 25% for each one. It was actually 42 and 41% for these outcomes, and 8 and 9% for these outcomes. And so what he noticed, and this was like after hundreds of flies were produced, what he noticed is that for some reason, the parental phenotypes, the combinations that mom or dad have, were way more common in the offspring than the recombinant phenotypes. You guys can see like these ones are a lot less common, or they're not occurring as often in the offspring versus these combinations, which is really weird because that should not be happening. Like it should be matching somewhat consistently. I mean, maybe not perfectly 25% for everyone, but it should be, give or take, due to random chance, somewhere close to that. And this seems like way off from that. And after repeating this many, many times, he kept getting similar results where these were way more common than these outcomes. And so when he figured out what was going on, through all his fancy thinking and modeling different ways this could be playing out, he figured out that these 2 genes that he was looking at, in this case for body color and the type of wing, those 2 genes must be on the same chromosome. And because they're on the same chromosome, it's going to mess up or it's going to then cause these alleles, the alleles for body color and the alleles for the type of wings, to not be completely independent of each other. Because now that they're on the same chromosome, during meiosis, when gametes are being made, that chromosome is those alleles are gonna travel together on the same chromosome. And I'll kind of draw this out and show you examples of this in a second. And that's going to cause certain combinations of alleles from this parent right here. Certain combinations of these letters or alleles are going to be more likely or favored in the gametes that are made than other combinations, because of the fact that some of these alleles are literally stuck together on the same chromosome. So when in meiosis, when they're lining up and being packaged into gamete cells, there's actually, if you're on the same chromosome, you're kind of traveling together into the gametes that are being produced. And so what's happening here is these gametes made by this parent, let's say it's dad, these sperm cells are for some reason being made way more common than these sperm cells is what's going on. These gametes are being made a lot more than these gametes by this fly, leading to these outcomes happening a lot more often. And so that's called gene linkage. That means that these genes are on the same chromosome. And so this picture might confuse you a little bit, but basically we're showing these alleles that this fly had on these chromosomes. So this is a gray fly with normal wings, but he's heterozygous for both, so on one chromosome. So if they're on the same chromosome, you can see here, like he has the dominant allele for body color, which is gray, and the dominant allele for wing type, which is these normal wings, on one of his chromosomes. But now on another chromosome, because he has homologous chromosomes, on the other chromosome, he has also those two genes, and he has the recessive alleles on that other chromosome, making him a dihybrid, right? He's big B, little b, big V, little v. He has, he's heterozygous for both traits because of the chromosome he got from his mom and his dad. Now, when these chromosomes when this cell, which contains these chromosomes, undergoes meiosis, these chromosomes are going to duplicate and then go through meiosis 1 and meiosis 2 and form these gametes. Now, when these gametes are formed, though, and these chromosomes get separated into these daughter cells, because these alleles are on the same chromosome, they're likely going to end up together in the resulting daughter cells that are made at the end of meiosis. Like this big B, which is on the same chromosome as the big V, those two dominant alleles are on the same chromosome. So they're likely going to stay together. Or like this recessive allele and that recessive allele, which are on the same chromosome, they're likely going to stay together. But it is possible that you could mix these up a little bit, because during meiosis, crossing over is going to occur when the homologous chromosomes are pairing up. And that is going to allow the opportunity for some of these alleles to switch spots. So you could end up with like this dominant allele being recombined with this recessive allele if there's crossing over that takes place during meiosis between them, and those can now you can end up with a new combination of DNA on this chromosome that puts these two alleles together and those two alleles together. But that's going to rely on crossing over happening in a specific way, which is going to make these outcomes possible, but not as likely as like the default. The default is going to be more likely because they're already together, ready to go. But every now and then crossing over will happen in a way that rearranges them, leading to these new combinations in the gametes that can be inherited or passed on to the offspring. And so that's why you see these numbers and the actual outcome a lot lower, because these are relying on crossing over taking place in dad's gametes, or when dad's cells are undergoing meiosis. So to kind of better visualize this, I'm going to walk you guys through an example that I'll kind of sketch out real quick with you guys. So if you look at, not this example, but let me pull up my camera. So I want to show you like what this would look like when genes are not linked, like when we're just looking at genes that are on different chromosomes versus when genes are linked and on the same chromosome. I want you guys to clearly see the difference, because this is a really hard concept to kind of grasp on a conceptual, visual level. A lot of students, it's hard to kind of understand what exactly is going on. So let me show you. So let's just start with, if genes were not linked. Like, let me show you how that would play out. Which is, in a lot of situations, they're not on the same chromosome. So unlinked genes means that we're looking at a situation where they're located on different chromosomes. So that's what it means to be an unlinked genes. The two genes that we're looking at are on different chromosomes. So on here, I'll even color code. So let's say we had an individual who was a dihybrid, like the fly that we were looking at, who's heterozygous for two traits. And for this, I'll just use A's and B's. So there's gene A, and they're heterozygous for gene A. And there's gene B, and they're heterozygous for gene B. Now, let's say that those two genes exist on different chromosomes. So what that would look like is you would have a chromosome in the cell, and on that chromosome would be let's say this is where gene A is. It's on this larger chromosome, and on one of those chromosomes it has the dominant allele, capital A, okay? But then on the other chromosome that I got from its other parent, so maybe this is from mom, and then there's another chromosome from dad, on that other chromosome is going to be the little a, because this person's heterozygous. So the gene A is on this chromosome, but it has two copies of this chromosome because it got one from mom, one from dad. It has a big A and a little a, okay? Now gene B, let's say gene B is on a different chromosome. Let's say it's on the smaller chromosome, okay? And on this smaller chromosome, let's say the chromosome from mom had a big B allele on it, and the one from dad had a little b allele on it, right? And so that would make this person heterozygous for gene B and heterozygous for gene A, right? And now they're on different chromosomes. So what's going to happen during meiosis, if I just quickly fill this in, is these chromosomes are going to duplicate, right? And so I could show each of these duplicated. So this chromosome is going to duplicate like this. This chromosome is going to duplicate like that. Now it's a duplicated chromosome made of two sister chromatids, duplicated chromosome made of two sister chromatids. And then same thing with these ones from dad. This one's going to duplicate. This one's going to duplicate. So now you have 4 duplicated chromosomes that have duplicated during interphase. All of that was during interphase. I'll even draw an arrow and write interphase. So this is before you started meiosis. You got to duplicate your chromosomes. That always happens first. First you duplicate them. And then in meiosis, kind of like what I showed here, they're going to pair up together. In meiosis, they find each other, the homologous chromosomes. So this chromosome that has gene A is going to find this other chromosome that has gene A, and they're going to pair up. And same thing with these guys that have gene B, they're going to pair up. And during meiosis, these homologous chromosomes, through 2 rounds of cell division, these chromosomes are going to the homologous chromosomes are going to separate in meiosis 1 and be packaged into daughter cells. And then the sister chromatids are going to be separated in the second round of cell division and form the final daughter cells at the end of meiosis. And so I'm just going to shortcut that quickly, because what's going to happen is, and hopefully you guys can visualize this by now in your head, what's going to happen in meiosis. Basically, if you have 4 chromosomes that you're starting with and they duplicated, at the end of meiosis, you should have now 4 cells that are produced at the end of meiosis, and they each should have 2 chromosomes that are now unduplicated. Once you separate the homologous chromosomes, and you separate the homologous chromosomes into 2 daughter cells, and then you separate the sister chromatids, you should now have 2 chromosomes in each one, each containing a single copy of each chromosome, the large one and the small one. The one that has gene A, the one that has gene B, which really, in reality, means that you're just taking one of these chromatids and joining it with one of these chromatids and packaging it into a daughter cell. And then so what we could do here is we could figure out all the different combinations in meiosis with all the different ways that these could line up and separate from each other into daughter cells. We could figure out right now, okay, what are all the ways that one of these guys could end up with one of those guys in a daughter cell? So for example, it's possible that like this large one that has the dominant allele for A ends up in this daughter cell, along with this small one that has the dominant allele for gene B. Which again, if you don't know why this is leading to that, you need to go and review meiosis. You need to go watch videos or look at pictures that we drew together or the graphic notes or random YouTube videos. You got to be able to visualize why this leads to this in meiosis, or could lead to this. Or it could lead to a different combination. Maybe you could end up with one of these one of these large chromosomes that has the dominant A. But during meiosis, it's also possible that that could end up with like the small chromosome that has the recessive allele like this. right? Like that should be possible too. Like one of these could end up with one of those, just like one of those can end up with one of these when these daughter cells are made. Or you could end up with this recessive A, a large chromosome that has the recessive A, like one of these guys, could end up with one of these guys that have the dominant B allele. And then lastly, there's one more combination that's possible. You could end up with one that has the large chromosome that has recessive A and the small chromosome that has recessive B. So these represent the 4 outcomes that are possible in meiosis. So in meiosis, all of these should be possible when meiosis happens. And you could stop the video right now and draw this all out if you want to, like and do meiosis and separate the homologous chromosomes, separate the sister chromatids. It should lead to every single time one of these, and when you make daughter cells, those daughter cells either are going to have that, this, or that. Okay, no matter how these line up or whatever, or even if you did some crossing over and you switch some letters, like it's still going to be one of these combinations in the end. And that's all the combinations, right? It's either this with that, or this with that, or that with this, or this with that. There you go. And so in meiosis, I'll even label this meiosis, you should end up with these 4 outcomes. Now, all of these outcomes should be equally possible. And if they're on different chromosomes, they will be equally possible. So when this person is making gametes, 1/4 of their gametes will be this, and this, and this, and this, because there's nothing really preventing any of these options from happening. This is all just very random how they're lining up and being separated. So you will get like 25% outcome for each of these being made. So when this person has a child, it's going to be an even chance that any of these combinations gets passed on. Okay, because again, they're a dihybrid. They could pass on a big A with a big B. That's possible. A big A with a little b. That's possible. A little a with a big B. That's possible. A little a with a little b. That's possible. And they're all equally possible as long as the genes are not linked. Okay, the law of independent assortment, any combination of alleles will be possible. This will match exactly what Mendel would have said would have happened. Now watch what happens when the genes are linked. Okay, now this one I'll go through a little faster now. You guys get the gist. Now in linked genes, this is now when they're located on the same chromosome. Now, when the genes are located on the same chromosome, let me show you what that looks like. So let's say, again, we have an individual who is a dihybrid. They're heterozygous for 2 different traits. Now, if they're on the same chromosome, let's say on this chromosome, there's gene A is on this chromosome, and let's say they have a dominant allele on it, and they got this from, let's say, dad, and then they got another one from mom, and on the one from mom, it has a recessive allele, like that. Right, so big A, little a. But on the same chromosome, this time, gene B is also on the same chromosome somewhere as well. And from this parent, let's say they got a dominant allele on this chromosome. So this parent passed on a big A with a big B on this chromosome that this person got. And on this other chromosome from the other parent, there was 2 recessive alleles, one for gene A and one for gene B. You guys with me? So now, like your big A, little a, one from each parent, and your big B, little b, one from each parent. But now they're on the same chromosome. Okay, this is what it means to have linked genes. Now, when this goes through meiosis, which again, we're just focusing on this chromosome. There could be other chromosomes. We don't care. We're just looking at the ones that have gene A and B on it. These chromosomes will first duplicate. So just like last time, you're going to go through interphase before you start meiosis. And during interphase, these chromosomes are going to duplicate themselves. So this chromosome from dad that has a big A and a big B, it's going to make a copy of itself. And now you're going to have that, 2 sister chromatids, exact copies of each other. And then same thing with this other chromosome. So now they've duplicated. You're now ready to start meiosis. And in meiosis, these chromosomes will find each other. These are homologous chromosomes. They have the same set of genes on them, the same set of genes on them. They're going to pair up along with all the other chromosomes that I'm not showing you. And then in meiosis, these chromosomes are going to separate into daughter cells, and then we're going to separate the sister chromatids into the final daughter cells, and you're going to end up with 4 daughter cells when this cell does meiosis. And so that's these guys here, either this guy, or this guy, or this guy, or this guy. In meiosis, And again, I really hope you guys are starting to be able to visualize what's happening in meiosis, where if you have two chromosomes that we're looking at, right, this one and this one, they're going to duplicate. And in meiosis, the daughter cells should now each have one copy of that chromosome. One of these, and one of those, one of these, one of those, of this chromosome. Like all of these guys are going to end up with one copy of this chromosome. And they're going to be unduplicated at the end of meiosis. Okay, these are now going to be haploid cells. Okay, there's not going to be homologous chromosomes anymore. And so if you guys look here, this is going to be a little different. So what could happen is this chromosome right here that has an A and a B, so basically one of these chromatids is going to end up in each of these daughter cells. Just like up here, one of these ended up in each daughter cells and one of these ended up in each daughter cell at the end of meiosis. Well, now since we're just looking at one pair, it's going to be one of these ends up in each of these. Okay, each carrying an allele for A and an allele for B. And so it could be this guy right here who has an A and a B, right? Like one of these guys could end up in here, right? Or it could be this guy over here that has a little a and a little b. In meiosis, that could end up in a daughter cell. But you guys can see that those alleles are kind of trapped together. Like this guy is with this guy, and this guy is with this guy, right? So they're going to kind of travel together during meiosis. However, technically it is possible to have a different combination than these two. And that will be possible, like for example, imagine, what if I told you it's also possible to have a chromosome end up in a daughter cell that has a big A, but not with a big B? What if I told you it's possible that this has a little b with it? And you might be thinking of how that's possible right now. Hopefully you are. That is possible because you could have crossing over taking place. Crossing over is going to take place between these homologous chromosomes, and as long as crossing over is happening somewhere between these two genes, and they're being switched somewhere in between, so somewhere in between gene A and B, they're switching, you could then have like those alleles switch, like let's say the B allele switch. And now this little b is with the big A and the big B is with the little a because of crossing over, as long as it's somewhere in between these two genes. If it's crossing over like below gene B or crossing over above gene A, that's not going to really help us. But if it's somewhere between, like somewhere in this area, if there's crossing over, switching that piece of the chromosome and that piece of the chromosome, or this piece of the chromosome, that piece of the chromosome, these letters will be carried with it, and we could swap them. And so you could end up with this, which means you could also end up with this, a little a with a big B, but that's going to again be dependent on crossing over happening in a very specific area on this chromosome. And so because these outcomes rely on crossing over taking place, which I'll even write a star right here, these two combinations are technically possible, but those gametes are only possible due to crossing over. So right here, I'm going to write some gametes, the ones with a star, which is like the big A with the little b, or the little a with the big B, are only possible with crossing over. Like that relies on crossing over happening in a specific way between these homologous chromosomes when they were pairing up during meiosis. And if crossing over didn't happen in that specific way, because there's going to be crossing over, and there's probably going to be lots of crossing over, but it needs to be a crossing over event that leads to these letters switching. And since you're relying on a very specific event taking place, it's going to make these less likely than those. Like these are just by default, you should end up with that, and you should end up with that and that. But these are relying on crossing over happening a specific way, which is going to equal these guys being less likely being made. Like they're not going to be made as often. Not to say that crossing over isn't happening all the time. It is, but the crossing over that leads to these guys ending up on opposite chromatids, that might not happen as often. And so what you'll end up with is more of the gametes, more than 25% of the gametes more than 25% will be this, and more than 25% will be this, because these should be the more common gametes that this person is making, because these don't rely on crossing over taking place. But these guys are going to be less than 25% and less than 25%. Not as common because they rely on crossing over taking place. So when this person has an offspring, it actually is going to turn out that it's more likely they pass on an A with a big A and a big B, or a little a with a little b, and no longer is it going to be equally likely that they pass on these combinations like up here. None of this relied on crossing over. You could have crossing over, you could not have crossing over. All of these would be equally likely because all of these are fair game as these chromosomes are lining up and being separated. You don't need crossing over to have any of these combinations possible. Whereas this is only possible if there's crossing over happening a specific way. Okay, and so now it's not as often is this parent going to pass on those two alleles or those two alleles, and more often they're going to be passing on those two alleles together or these two alleles together, because the genes are linked and these alleles are going to be traveling together when gametes are being made and passed on together in preferred combinations to the offspring. And so if I go back to the slides now, that's exactly what's going on. Hopefully you guys see now. That's exactly what was going on with these two combinations here from this person who was a dihybrid, which is the exact same example I was showing you in that picture I drew out with the A's and the B's. Big A, big B, little b. These combinations are more likely to be passed on than these new recombinant combinations. But those recombinant combinations are possible, but it's going to rely on crossing over. And so anyway, so then that alone is a pretty big discovery. So anytime, I'll just tell you guys right now, anytime you have a cross like this, it's usually going to be a cross between a dihybrid and a homozygous recessive individual. That usually is a cross that will show you if the genes are linked or not. And if you cross a dihybrid with a homozygous recessive individual and you get outcomes that show the parental phenotypes are way more common than the recombinant phenotypes, which should not be the case if everything was if these alleles were completely independent of each other and there was the law of independent assortment was working out the way it was supposed to. But if that's not happening, if these are more likely than those combinations, that can best be explained and usually always be explained by those two genes being linked. That must mean that's evidence that those two genes are on the same chromosome. That's the key takeaway here. Okay, if you guys see a situation like this where certain outcomes, specifically these parental phenotypes, are occurring way more common than they should be, and these other outcomes are occurring way less often than they should be, gene linkage is the answer. Okay, that's what's going to mess this up, as I kind of illustrated on that little picture showing you how in meiosis there is going to be favoritism for some of these combinations when they're on the same chromosome. So at the very least, just memorize that. Even if you don't fully visualize why, or it's kind of confusing you why it's working out that way, just know that that's the explanation here. Now, to take it a step further, he actually started using this data that he was collecting to actually start mapping and figuring out how far genes are away from each other on a chromosome compared to other genes. So he actually started doing something called chromosome mapping, which is where you actually start figuring out what distance genes are from each other on a chromosome. And it's dependent, what he found out is that it's dependent on something called the recombination frequency, or the recombinant frequency. The recombination frequency is the total percentage of recombinant phenotypes that are produced in a cross between a dihybrid and a recessive individual, like this cross I just showed you. The recombinants are these guys, and the recombination frequency would be the total percent of offspring that have this recombinant phenotype, which is this phenotype and this phenotype. So it's 8% and 9%, so total that's 17%. So the recombination frequency would be 17% for these two genes. These two genes, it looks like they're recombining in these new combinations 17% of the time. That's the recombination frequency, which is what I have listed right here. You can see it was 17% for those two genes, for body color and the type of wing. Now that recombination frequency is actually directly related to how close and far away the genes are. The closer the genes are on a chromosome, the smaller that number is going to become. And that's because the closer the genes get together, the less likely that crossing over is going to be able to separate them from each other. But the further they're away from each other, the more likely crossing over will be able to happen somewhere between them and move them and switch them into new combinations. And so visually, just to make sure you guys see what I'm talking about here, on here, if you guys look, okay, if as you move the A and the B gene closer together, okay, if these genes are closer together, it's going to start becoming less likely that crossing over is going to happen somewhere between them, switching these alleles, which is going to make these guys occur less and less often the closer these genes are. And so what you're going to see happening in the offspring is the recombinant phenotypes that are made because of these gametes, they're going to be occurring less and less and less. But if you have a lot more recombinant phenotypes occurring, that's telling you that they're further away from each other. So Thomas Hunt Morgan figured this out, that by looking at the recombinant offspring, that actually tells you or gives you an idea of how close those two genes are. And so what you guys just need to memorize and hopefully kind of conceptually understand is that the closer the genes are together, the less often crossing over is going to happen between them, which means the smaller the recombination frequency is going to be. You're going to have less offspring produced that are created from these gametes. But the opposite is true then also. The further away the genes are, the more likely crossing over is going to happen, which is going to make these gametes and these recombinant phenotypes in the offspring more popular. Okay, so if this number was large, if these percentages were larger, that suggests that the genes are further away. But if these percentages are smaller, that suggests the genes are closer together, because these are becoming, the less likely these are, the closer the genes have to be together, which is preventing crossing over from separating them. But the further they are on a chromosome from each other, the more likely these recombinant phenotypes can take place. And so he actually came up with what's called a map unit. A map unit is a unit of measurement we use on a chromosome, which is basically just the recombination frequency. So one map unit equals 1% recombination frequency. So for like the genes for body color and whether or not you have vestigial or normal wings, so like the wing gene and the body color gene, we say that they're 17 map units away from each other. And that's because the recombination frequency between them was 17%, as shown and demonstrated in his experiment. 17% of the time you got these recombinant phenotypes. And so he is going to say that that means that they're 17 map units away from each other. Now these units are just all relative. And so you could compare that to like how many map units they are from other genes. So like, but if you look here, you can see for this other gene, this is the eye color gene and the body color gene. The recombination frequency between them was 9%. So only 9% of the total offspring, when they did this special type of cross, had a recombinant phenotype. And so that means that those two genes are 9 map units away from each other, which means they're going to be relatively closer together than the body color gene and the wing type gene, which were 17 map units away from each other. So these guys had a 17% recombination frequency, which puts them at a certain distance away from each other. But when these two genes lead to a even smaller recombination frequency, that tells us that they must be even closer together. And so in your notes, there's a couple examples where you guys can look at the recombination frequencies and start to figure out if those are the recombination frequencies between these different pairs of genes. You can actually start figuring out the placement relatively of where these genes are and which genes are closer and which genes are further away. If they have if two genes have a large recombination frequency, that means that they're probably far away because crossing over is happening a lot between them, making these recombinant offspring. But if they have a small recombination frequency, that means the genes must be closer together and crossing over is happening less commonly between them. It's not happening as often in a way that mixes those alleles up, making less recombinant offspring. So that's another concept you guys need to memorize, but hopefully conceptually understand a little bit. And then lastly on these notes, which again, I know this is a lot of explanation here. It's a long video. But the last thing on here is using a chi-square test to determine if genes are linked. And so you guys will see a lot of questions, a lot of AP type questions where they ask you to do a chi-square test to see if a certain cross is producing a set of offspring that match what we might expect to happen, or if it's producing a set of offspring that doesn't match what we would expect to happen in terms of their phenotypes that are occurring. And so one way we could use a chi-square test is to see if genes are linked. Because for example, if I go back to this data here, right here where the actual outcome was 42, 41, 8, and 9, when it was supposed to be 25, 25, 25, 25. So you might argue like, well, how do you know it's not just because of random chance? Because it's not always going to be perfectly 25, 25, 25, 25. By random chance, the actual percentages can vary. And so someone might argue, well, maybe this has nothing to do with the genes being linked. Maybe just by random chance, these guys were occurring more often than these guys, by random chance, like nothing to do with anything else. And so that's a fair argument, sure, like someone could say that. But then what we could do is do a chi-square test to see if this is actually a statistically significant difference in the actual outcome compared to the expected outcome. And so that's what we use a chi-square test for, like we did last semester, is we use it to see if the actual outcomes that you're getting are statistically significantly different than the expected outcome. And if they are, then you can now have stronger evidence that, yeah, this is actually a real difference. It's not due to random chance. So let me show you guys how to do that with this example actually from your guys' notes, the data I just showed you. We're going to see if there's a statistically significant difference between the data that Thomas Hunt Morgan collected versus what Mendel would have expected to happen. And so here's the formula. On the notes, you guys, it has the formula to remind you how to do chi-squared and the chi-square table. But I'll finish this video by doing an example, the same example that's in your notes, actually. And so if you guys want to work out this question with me, it's in your notes. Let me pull up my camera and we'll take a look at it. But basically, it gives you guys... I printed a larger version of it, but it basically gives you the real data that Thomas Hunt Morgan collected when he did his cross. And so again, this was from the dihybrid cross with the fly that was a dihybrid and the recessive fly. Okay, this cross right here. And so this was the data he actually collected. These are the number of flies that had these different phenotype combinations. These guys were the parental phenotypes, like that was just like dad and that was just like mom. And these were the recombinant phenotypes here. But like I showed you earlier in the slides, you guys, when you do this cross and you set it up, which I'm not going to set it up again, go look at the slides if you don't know how to set this up. When you set up this cross and you figure out there's 4 things that this parent could pass on and there's only 1 combination that this parent could pass on, your expected outcome should be 25%, 25%, 25%, 25%. And so you guys, for your expected outcomes, you're always going to use the outcome that would be predicted by simple Mendelian genetics. Like if everything just works out the way that Mendel said it would work out, and there was the law of independent assortment, and any of these combination of alleles were equally likely to pass on, that's always going to be what you guys expect, which means that's always going to be your null hypothesis for a chi-square test. Your null hypothesis should be whatever normal genetics would have predicted or expected. So for your null hypothesis, don't say for your null hypothesis that the genes are linked. Like that's never going to be our null hypothesis. The null hypothesis is just going to be by default, if everything was happening the way that has already been proven to be determined and a normal thing, and there's no craziness happening or nothing super exciting happening, that's going to be your null hypothesis. And for this case, Mendel already showed us how simple genetics should work, and so there should be an expected outcome here if everything's working out in a normal genetics way with the law of independent assortment and everything. And so you should get, so for my null hypothesis, I'm going to say a cross between flies with these 2 genotypes will produce offspring that are 25% this, that, and this, or a 1-to-1 to 1-to-1 phenotypic ratio, which if you do the Punnett square, that's exactly what you should expect to happen. That should be your null hypothesis, which basically you're just assuming that these genes are not linked. Like we're just assuming that these genes are linked. We're going to assume a situation like this, where like all of these should be equally likely to be passed on from this parent, producing these phenotypes in the offspring. Now, to do a chi-square test, then we could see if this varies statistically significantly from that expected prediction. And so you guys would have to first calculate the expected numbers. There's a total of 2,300 flies. So to figure out the expected numbers, since we're saying it's a 1-to-1-to-1 ratio, we're going to take 25% of this number. That should be our expected number. So you would do 2,300 times 0.25, like what's 25% of that, which luckily I have a calculator right here because I don't know what this is. That's 575. And that's how many should be for each of these if everything's working out the way that Mendel said this would work out, which would be a total of 2,300 flies, right? But we were expecting this proportion in the outcome, whereas in reality, we got this right here. And so what we got to do, like I said, is we got to prove that this is not just random chance, okay? Because someone could argue like, well, actually, I think that this doesn't necessarily mean that it's not a 1-to-1-to-1-to-1 probability. Maybe this is just random chance that we got these weird numbers. Well, let's find out. And so to do a chi-square test, here's the chi-squared formula. You would do observed minus expected squared divided by the expected. And then I forgot the sigma sign. This is on your guys' formula sheet, and I put it on the notes just to remind you guys. So there's the formula. Right, so the way we plug this in, hopefully you guys know how to do this from last semester, but I'll do it real quick. So for outcome number 1, being gray with normal wings, we observed there was 965. Oops. But we were expecting 575. So you're going to subtract those numbers and square it and then divide it by 575. And then you're going to add that to the next possible outcome, which was black with vestigial wings. And so go ahead and do that for all 4. Or you can watch me do it for all 4. And for the next outcome, we observed that there was 185. We were expecting 575. You subtract it, you square it, you divide by the expected. And then for the last outcome, we observed 206. We were expecting 575. You're going to square it and divide by the expected. So that's how you guys should set up the chi-squared formula when you plug in these numbers. And so you're going to have some really big numbers here, I can already tell you. And so see what you guys come up with when you do all of that. I'm going to just do it all on my calculator right now and then tell you the final answer. And hopefully you could do the math from this point to get to the correct answer. Just make sure you guys subtract and then square it before you divide and add the next number. So feel free to do it in baby steps if you need to. Let's see if I can plug this in without messing up, because I don't have the answer written down yet. Okay, I know the suspense is killing you. If I plug this in correctly, which who knows, but I got for my chi-squared value 1002.65. I'll just round it 2 decimal places. It's gigantic. That's a large chi-squared value, which sounds right. It should be a very big chi-squared value because this is pretty solid data here. Like, this does seem pretty different from this data. This wasn't just a few flies. It would be hard to say this is random chance when we're making 2,300 flies. Anyway, you get a large chi-squared value. Now here's what that means. So once you guys get your chi-squared value, which I really hope this is correct or this is embarrassing, but I'm committed to it at this point. It should be a big number. Now you're going to go to a chi-square table, just to remind you, and you're going to find the critical value to compare this chi-squared value to. And so to do that, you need to know the degrees of freedom. For this question, the degrees of freedom equals 3, because it's the number of possible outcomes minus 1. So degrees of freedom equals 3, just to remind you. So when you pull up a chi-square table, which I have one in the notes conveniently right here, you're going to look here at the chi-square table and look at 3 degrees of freedom, which is right here. And you're going to look at these are the critical values. Okay, there's one for a p-value of 0.05 and one for a p-value of 0.01. These are your significant levels. Usually, like I told you guys, you can just use the one for a p-value of 0.05 unless you're told otherwise. But that would give you 95% certainty that your data is statistically significantly different than what you expected to happen. Or if it's bigger than this other p-value, that would give you 99% certainty that, yeah, this data is weird and doesn't match what was expected to happen. And you guys can see that our value is way larger than both these values, both critical values here. And so what that means is, since our chi-squared value is larger than the critical value for 3 degrees of freedom and a p-value of, let's just default to 0.05, that means, you guys, that we can reject our null hypothesis. Our null hypothesis, which I wrote up here, I said our null hypothesis, like the reason I got these expected numbers, according to my null hypothesis, I was expecting there to be a 25%, 25%, 25%, 25% outcome in the phenotypic ratio for the offspring. And that's why I put these numbers here, because it matches my null hypothesis. And what we can do now is we can reject this now, because our chi-squared value is larger than that, which means this is not correct. Like, we have data that says, that supports that this is not the correct prediction. Like, this does not match with what was actually happening. There is a statistically significant difference between what happened versus what we were expecting to happen. And so at that point, you could come up with some kind of alternative hypothesis or alternative explanation. Maybe at that point we would say, okay, well, maybe it's because the genes are linked, which would definitely be the best explanation at this point. The genes are linked. And this is not following the law of independent assortment. And I have a chi-square test and statistical significance to prove it. Now, if this value did not exceed the critical value, if our chi-squared value is less than the critical value, then we would accept the null hypothesis, or we would fail to reject it, which means that we would say, okay, our data matches well enough with what was supposed to happen. There's not a statistically significant difference. And it's probably just due to random chance that is varying. But in this case, this is definitely weird. Like, this does not match that, okay? So that's why we're going to reject the null hypothesis. Anyway, that's it for this long video, but I hope that helps a lot. We'll talk a lot more about this in class and do a lot more practice. And I'll see you guys later.